A very warm welcome to our channel, children. So today I'm here to speak about linkage. Linkage, as you everybody know that this is the most important topic when coming to 12th biology. Now here when we talk about linkage, we already know that this particular concept is present related to genetics. So when you talk about genetics, everyone knows that, sorry for the hoarse voice, but for genetics, it's a branch of science that deals with heredity and variations. So when coming to heredity, we all know that all the genetic characters are inherited from the parents to the next generation, that is nothing but to the offsprings, and these variations that we are talking about is because of the crossing over. So when we just see the definition of genetics itself, it says that it's a study of, or it's a branch of science that deals with heredity and variations. Heredity is because of the linkage, and this the variations whatever we are observing in next generations or the preceding generations is because only of the crossing overs. So today as this topic we are getting more of questions from in the NEET examinations as well as in the CBSE board biology. So today what we are going to do is we are going to see only a little bit concept of this, how the questions, we are just going into the topic right now. So let's dive into the topic. So the first one what we are going to do is studying the definition of linkage. So the word linkage itself, when we are hearing the word itself, we will understand that's a common word, everyone knows that. So link, whenever we are talking about linkage, it just says that something is linked over here. And what is linked? As we are studying about genetics, the genes are linked together. So when genes are linked together, everyone knows that, because this is only a revision part. Now what we are talking about is genes are located on the chromosomes, and on the chromosomes they are just not located here and there, they do have certain positions, and these positions or locations are called as locus. Locus, when we talk about, this is a word which we don't mention while writing in the examinations, because keywords are more important for scoring marks. We just write that on the chromosomes the what is called as the genes are located, but we don't use such type of words. So better, genes are located on a certain positions on the chromosome, and these positions you are calling it as locus. So in plural we use another word for locus, that is nothing but the loci. So loci is the positions when we are talking about more genes. When we talk about a single gene, we are calling it as locus. So this is what introduction of your locus and genes. Now moving on, when these linkage, when we talk about, we are talking about the linked genes. When linked genes, when we talk about, linked genes are nothing but one or two genes or two or more genes, when they are arranged onto a particular positions in a chromosome, they are inherited together. So they do have their own identity, but in generally when we talk about, these genes are located near, or the distance between these two genes are very lesser, hence we call them as linked genes. So here two types of linkages we do have, that is nothing but the complete linkage, and another one is the incomplete linkage. So before we dive into that particular topics, we'll just have a quick what is called as the definition of the linkage. So genetic linkage is the tendency of genes, as already discussed about, I've given you all the information about genes now. So genetic linkage is the tendency of genes that are located close to each other. As I already told you, when they are arranged close to each other, the distance between them is very lesser. Based on this distance only, we are dividing them into non-linked gene and the linked gene. So distance between them is very lesser, we can say it's small on a chromosome, and these genes are inherited together. We know that what is the process, what is that cell division in which the genes are actually inherited to the next. We know about the gametes. Now here what usually happens over there, what is that process? We are calling that division as meiosis. So during meiosis, this linked genes are inherited to the next particular cells, which we are calling it as the gametes. Now genes, all this is also I discussed you before, genes are located on the locus. Loci, the word when they are using it for plural. So are nearer to each other, are less likely to be separated, they cannot be separated, as already mentioned, they cannot be separated. When they are inherited from one generation to next generation also, they will be inherited together. So now here, or we can say that they are inherited as a whole. So different chromatids during chromosomal crossover. Now coming to the crossover word, What is crossover? Crossover is nothing but a word which is nothing but a cellular process in which the homologous chromosomes. Now what are homologous chromosomes? Homologous chromosomes, when we talk about at the time of meiotic divisions, these homologous chromosomes are situated near to each other, and then we already know that one chromosome will be getting from the paternal side, one chromosome usually get from the maternal side, and when they line up together, they exchange the arms. And this particular process where the crossing over is taking place, you are calling it as here they are genetically linked. So now moving on to the next one. So I'll just go into the brief introduction here. So when we are talking about the chromosomes, for example, this is a normal structure, is not a typical one, but in generally when you're talking about the locations, so when the genes are located far away to each other, you are calling them as, so for example, this is a gene, this is also a gene, they are having a further amount of distance over there. Now because of these distance, we are calling such type of genes as non-linked genes, but these are present on the same chromosome. They are not, I mean not talking about the different chromosomes. On the same chromosome, distance is also bigger. So what we are able to understand is the linkage. Now the linkage is always dependent on the distance. So we can also give you, I'll give you a small what is called as the definition. It's not like, remember this is in the form of an equation. So linkage frequency is always inversely proportional to the distance between the genes. So what we are able to understand over here is, as it is inversely proportional, it is not directly proportional. Directly when we just say is distance also increases, the linkage or the linked genes also they come closer, but it's not like that. As it is inversely proportional, distance between the genes is higher. If it is higher, then linkage or the linked genes or the linkage between the genes will be lesser. So it is actually inversely proportional. So now coming on to the chromosomes, as we have already discussed about the linked and non-linked genes, I have already given you the linkage frequency formula. That is also more important when coming to the competency based questions in most of your objective based. So they will be asking question based on the recombination frequency as well as your linkage frequency. So this is the basic thing what we have to remember. Now coming on to even more deeper, it's not like a very big concept to remember, it's a small one, but based on this many questions have been asked, numericals also has been asked. So we have to be prepared. When we just miss out these small gaps, the small gaps cannot be left over, because these small gaps, whatever we leave at the time of preparation, that only will lead to a big blunder. So now coming on to the linkages. Now here when we are talking about the genes, for example, this is a chromosome arm. Now the genes which are located very near, so distance, as we have already discussed, the distance was larger in between the locus of the arrangement of the genes. Now here the distance is very lesser, they are very close to each other. Now we will see the inheritance. So such type of genes cannot be separated. We have already studied about a law before. We'll discuss that law later, but here when these genes are arranged very closely to each other, they will be inherited as a whole. So you cannot separate this. So they will be inherited. Inheritance, the word itself says that carrying the information from the parents to the next generation or to the next offspring. So they will be inherited as whole. Now the question comes, which has been asked in most of the board examinations. It's a 1 mark or like mostly coming in assertion reasoning questions only. Now here what is the question which has been asked 2 or 3 times. Now here what they are asking is how many number of linked genes or linked genes are present. That's the question is being asked. They'll give you some sort of number and you have to find it out, the number of linked genes, and at that time we usually get confused. Now what's the major point over here is we have to remember this is a complete thing which has to be stamped over here in your mind. Now when they are asking how many linked genes are present, so number of linked genes number of linked genes is always equal to the number of pair of chromosomes. Number of pair of chromosomes. When I am talking about pair of chromosomes, I am not talking about the total number. For example, if you take human beings, we are diploid organisms. We are diploid in the sense 2n. N is visually denoted for it's a haploid. So now as we are diploid, we do have 46 chromosomes. I am not talking 46 pairs, it is 46 chromosomes. Now if it is 46, now here you just see the question, how many linked genes are present? They have not asked you about human beings, but in generally, how many linked genes are present? Then we usually say that we know that it is 23 pairs. So 23 will be your haploid condition. So here, as it is 2n, now this one will be your 23. So how many? We do have linked genes. So we talk about that. Number of linked genes in human beings is 23, not 46. So this will be the answer. So when they are asking such type of questions, we should not think about the total count. We have to think about the haploid set only. So number of linked genes is always, we are talking about the haploids, and that is the reason these 23 pairs of the chromosomes which are present in a human body are inherited as a whole to the next generation. This is what we have to discuss. So as we all know, the law of independent assortment, which has been given by Gregor Johann Mendel, who was the scientist who has been given you the different laws, law of dominant, law of independent assortment, law of segregation. Now here when we come across this linkage or linked genes, what is the law which he has explained? It was law of independent assortment. Now what do you think that law is applicable over here? No. Why? Because there what he has explained is the genes which are, because he has taken the consideration of the green pea plant, that is nothing but the Pisum sativum. So he has taken that plant and he explained that each and every gene has their own characters and they are inherited or they have assorted at the time of the production of the next offspring. But here when we talk about the what is called as human beings or any other examples, we just say that when coming to the linked genes, what is not applicable? A law of independent assortment is not accepted, and that is the reason this particular law was not accepted worldwide. And that what is the major reason behind this is the linked genes. We will take one more question in which I will explain you what is recombination. So whenever we are talking about variations, when like for certain amount of chromosomes which are inherited from the parents to the next generation, we usually see there is a new particular characters has been like inherited to the next generation, that is nothing but the young ones. Now how actually this is being happening? That is the reason for the evolution as well. Now here when we talk about what is called as the recombination, one question which is being repeated most of the time after the what is called as in NEET examinations, they have asked a step, they will give you a particular flowchart and from that you have to identify the answer. But before that we should know the percentages. Now coming on to, we will just take a simple example to explain the recombination. Now I will take a set of chromosomes here. So here I am talking about the non-linked genes, I am not talking about the linked genes. So this one is non-linked and this one will be, I will take a linked gene here. So we do take a particular set. When it is non-linked, take it as capital A, capital B, the distance should be longer, and here we will take small a and small b. I am talking about homologous. When I am talking about homologous, I have already explained you, but furthermore I will explain you later. So here this is non-linked, both the genes are having certain amount of distance between them in the locus. Now coming to the linked genes, here the distance between the genes is smaller, the distance between the gene is smaller. Now coming on to the further production of the next offspring. So how we can see the possibilities of the genes now, because after the crossing over we see certain type of variations. Now you can see over there, we will take particular what is called as combinations. You combine with this everybody I would have done with the wild type, which is nothing but which is being done by Morgan in the fruit fly or Drosophila melanogaster. We have already that picture in the textbook, you know that, but only for your easy understanding, we'll just make it like this so you can understand the percentages of recombination. So A, here 1 will be the parent type because this is the parent gamete. So here 1 will be the parent type, another one you will have the combination of the small b. Now here we'll take small a, capital B, and another one will be the parent, that is small a and small b. Now how many recombinant species or how many recombinant genotypes you have observed this one is the parent, this one is the parent, and you are having two particular combination or recombinant frequencies here. Now we just usually give them a number, we cannot sit and count what is the percentages, but in generally we assume a percentage for this because this is the property where recombination takes place more. As crossing over is taking place, recombination also will be high. Now coming on both of here, we usually take 48% for this particular genotype and 48% for this particular genotype, and what is left over 4%. So we will take that, either a genotype may be of one of the parent or the genotype of the another parent. Now here what you have observed is the parental gene frequency or the parental gene percentage is lesser, parental gene percentage is lesser, but the recombinant is nothing but the new particular genotype what you have got. So here the recombinant gene percentage is higher. So what you have understood now, the final point is, in non-linked genes most of the genotype will be looking similar, or we cannot say looking because we are not talking about the phenotype. Anyhow genotype, if it changes, it will show differences in the phenotype as well. Now here in non-linked genes the recombination percentages will be higher. Now coming to the linked genes, here, as the distance again will make 4 particular possibilities of genotypes. The first one, what you have to do is the simple percentages, but we have to draw it little bit closer. Now when we talk about one will be like similar to that of the parent, another one we have to take a new combination, that is nothing but again the same thing. Here you will take the same thing again, and finally you will be having the parental type. Now all the 4 genotypes are closely linked together, hence we are calling them as linked genes, but when you observe over there, it's only the assumption. Now you will be talking about that the 48% will be more similar to that of the parent, one of the parent, and then here also, and the remaining 4%, because no crossing over is taking place over here, possibly not, we cannot say that it's not completely no crossing, percentage will be lesser. So here you will be getting it out 2% and 2%. Now what you have observed after this conclusion, if you get these points properly, you can easily solve the questions which has been asked. So what you are having over here, this is in this particular linked genes, they are similarly, or we can say that they are having the recombination frequency which is lesser, and the parental gene or the parental gene genotypes, they will be having more. So this is final output what we are talking about the recombination as well as. So what you have understood finally about this is, here the linked and non-linked, when we are talking about crossing over, we are talking about the variations which have been seen as well as the recombination or recombinants. Now we are going to see that, because I already learnt about the recombination frequency. This is the question which has been asked in the NEET examination as well. Now they will be giving you numericals based on this. Numericals will further see in the next particular video, but here what you are going to do is see the formula once, so you will get an idea. Now what is the formula of recombination frequency? Now you know that how many progenies you have got. You have seen the roughly what I have made you the explanation over there. You have seen that how many recombinant frequencies or how many recombinant what is called as offsprings you have got. So calculate that progeny. Now finally they will give you the number. You need not sit and count the complete crossing overs over there. They will give you certain numbers and you have to find out. For finding that, we do have a certain what is called as the formula. Now recombinant frequency is nothing but number of recombinant offspring. So we have seen that 2 parent offsprings, one of the parent offsprings we had which looks similar, or which we can say that they are having the genotype similar to that of the parent. and we have got certain type, that is we are talking about the 48% of recombinants which we have been already formed. So you have to take that count, number of recombinant offsprings, how many are there, they will give a certain number, you fill it up over there, and upon, that is nothing but upon total number of offspring, how many offsprings, the number which they have given into 100. So that will be giving you the final recombination frequency. So this is all about what you are calling it as a linkage and crossover, not so deep. Even this topic can be explained even more deeper by taking certain more examples, but these are the basic things what is needed for writing definitions, we can tell you about small and very short questions. So there you can write the definitions of all these things, and any numerical if they have been given to you, you can certainly go with this particular type of equations. So I hope you liked the video. For further more videos, please do subscribe our channel, and you can further dive into even more educational videos. Thank you so much.